On Web CT, you have the extra credit there. It's for each respective test, right? 5 points.
That's on the. Those are outdated study guides, as I mentioned at the end of the semester.
Okay.
What I'm hoping to be able to do is to generate a couple of quizzes that would serve the same purpose. It won't happen for the first exam.
Okay. Thank you.
But they're still viable study guides, just the same, yeah. What we'll need to do is to pick up where we left off with Lecture 6 on gastrulation and then go on to Lecture 7. I unfortunately did not get a chance to record the lectures, but I'll work on it through into next week, so that we have all the lecture material available, either from in-class or from the pre-recorded. There's my eyewitness for having to go to the hospital. She checked us in for my son to get his blood drawn. And everything's fine with him now. He turned out to have low theophylline levels and at no risk of any type of cardioactive impact. It was a little worrisome for a while there, but everything seems to be back on track. The notochord is the focus of our attention right now because we are going to shift a little bit to neurulation. Again, I remind you, we are not at the point of studying organogenesis. This is still morphogenesis. The conceptus is taking structure, and we want to understand and appreciate some of the basic milestones that give all the axes. Dorsal-ventral we discussed already, that was amnion to yolk sac. Rostral-caudal, that was prochordal plate to the primitive streak. And obviously right and left as soon as we split things down the middle, and especially one of the prominent midline structures extending from the mid-axis basically of the entire embryonic disc toward the prochordal plate is the notochord. We recall that all of the cells that we are discussing forming the three-layered embryo and the events going on localized within that originate from the epiblast cells. So as epiblast cells differentiate and lose some of their pluripotency even, they're somewhat remote from totipotent, as you recall. They're going to migrate or be pushed in a column and be migrating everything together. There's obviously different types of migration, extending protoplasmic processes and moving like that as we think of the amoeboid type of movement, navigation through the extracellular matrix. And then there's just a mass of cells that are growing and dividing and creating a path for themselves to move throughout. And the notochord is that type of a solid column of cells with the division within it displacing cells and extending it in a single direction in this case. It doesn't migrate laterally. So something tells those cells not to have any type of migratory pattern extending from the delimitations of the notochord. So obviously a tremendously orchestrated intercellular, there's a lot of intracellular communication going on as well, obviously, but intercellular communication either through the physical contact of the cells or their interaction with the extracellular matrix or through extracellular chemical messenger systems. As these various clusters of cells migrate away from the cells of origin, sometimes the cells of origin now take on a new name like the ectoderm that differentiates into surface ectoderm and neuroectoderm, as we will see, and the mesoderm and endoderm. And here we have this notochord that is basically a signaling mechanism, as we will see. And therefore, it leads to the initiation of the differentiation of the overlying ectoderm into the neuroectoderm, and therefore what doesn't become neuroectoderm is surface ectoderm to differentiate into various structures. So we had seen this previously at the primitive pit. There's this column of cells that begins to divide and burrow down into the mesodermal layer, unidirectional toward the prochordal plate rostrally. And as it extends, we see in sagittal view and then cross-section, transverse section in these parallel panels C and D. As it extends, it begins to become a cylindrical structure rather than a solid core of cells. It's preceded by a solid core at its most rostral end. The older portion, if you will, loses the cell content within it, so it is not a solid core. It is an empty cylindrical-like structure. And ultimately, that opening extends to the rostral portion of the notochord. Then, as we had stated, there is an opening that occurs, the neuroenteric canal, the one time at the amniotic cavity and the yolk sac cavity are continuous. In addition, there is this opening of the notochord so that it's no longer cylindrical, and it becomes intercalated, as stated here, in the embryonic endoderm. So it does extend past the mesoderm to interact with the endoderm. And as this proceeds, the mesoderm is going to begin to differentiate and be laid down as the paraxial, intermediate, and lateral intraembryonic or just embryonic mesoderm. Primary reason for continuing through these series of slides is so that we can see these illustrations. The utility of these is obvious. So the notochord is present just adjacent to the mesoderm, and of course this actually would be the paraxial mesoderm, but we're not being that definitive or detailed in this illustration. The overlying remaining epiblast cells are, of course, now the ectoderm. Very soon, just above or dorsal to the notochord, there will be a stimulus by the notochord for these cells to differentiate into neuroectoderm, and then the rest becomes the surface ectoderm. But for now, we'll refer to it as ectoderm. And then, of course, quite obviously, the ventralmost layer or collection of cells is the endoderm. So we have established the three-layered disc, ectoderm, mesoderm, endoderm. This is well established as the embryo now with the three germ cell layers. So the space on, ventral to the embryonic disc is the yolk sac, the structure in its entirety. Sometimes just the term sac is used for the cavity as well, but we remember that we think of a sac actually as having the structure that contains it, the paper bag, if you will, and then the space within it that can hold things, the cavity. This slide then is from another view. What is this view? How would you describe this view of the embryonic disc? Ostrocaudal, transection, or transverse section. Dorsal view. So it is the dorsal view. We're looking at the cut margin of the amniotic sac, and we see the primitive streak, the primitive node, and then in a ghost-like fashion, looking through the ectoderm, the notochord. So this other point, the future cloacal membrane, is another example of where epiblasts and hypoblasts sustain their tight junctions with each other, and no mesoderm or any other type of cell can interpose between those two layers. So in reality, this is the caudal pole established by the future cloacal membrane that will degenerate ultimately to form the anus and some other structures for the urogenital system, as we'll describe later. And then the prochordal plate we've described as becoming, actually will be the buccopharyngeal membrane, ultimately separating the oral cavity from the oral pharynx way at the back of the tongue. So the prochordal plate, the notochordal process, the primitive streak, primitive node, and the future cloacal membrane are shown on this dorsal view of the embryonic disc. What view is this? Transverse, dorsal, or sagittal? Sagittal view, right? So it is cut from prochordal plate to future cloacal membrane, if you will, and we've separated it into two halves, right and left, and we're looking at the cut edge of either the right or the left side. They, of course, would be mirror images. That is, of course, the symmetry that we're all familiar with. So immediately we recognize where the primitive pit had been located. It's kind of a different structure now. It's not just a pit. It's kind of become a sinkhole, if you will, extending all the way through the notochordal process. We apparently have lost the number 4 to go with that label there. No, it isn't 4. It's 5, yeah, sorry. So we recognize the dorsal aspect for being formed by the yolk sac with its cavity, and not the yolk sac, the amniotic sac, sorry, and then the ventral aspect by the yolk sac and its cavity. Immediately we recognize an extension caudally. We know the notochord is projecting rostrally. Prochordal plate is going to be there at number 4. And the future cloacal membrane is going to be located here caudally. So at the caudalmost portion, or the caudal pole of the embryonic disc at its junction with the secondary yolk sac, we see an extension of the endoderm into the structure that is now securing this embryo with its attached sacs to the chorion. Let's just use that collective term. And that's the body stalk. So this is going to become, obviously, the umbilical cord ultimately. Right now it's just the body stalk. It doesn't have the vasculature and appropriate connective tissue and surrounding on it just yet. This, of course, is related to urinary type of excretion in avian species, at least. We're not quite certain what it is, what function it may be performing in mammals and in humans, but it does form just the same, and this is the allantois, A-L-L-A-N-T-O-I-S, of course, not listed here, and it's a distractor label, right? So one would be obviously the amniotic cavity. We use that term specifically in this case. The surface ectoderm, former pure epiblast, that now is the surface ectoderm as it's given rise to all the other germ cell layers. 3, of course, is still going to be the primitive pit. 4 should be the prochordal plate. Then 5, the number missing, would be the notochordal process. And then we know that extending at the midline, where we know this is a median sagittal section, so a mid-sagittal section, because we can see the continuity of the opening through the primitive pit right into the inside, now empty portion of the notochordal process. So that is telling us we are directly midline, mid-sagittal. So immediately caudal to the primitive pit is the primitive streak. So in the previous series of slides, we saw that the prochordal plate in this area, the notochordal process these ventralmost cells at its rim will interact and interpose themselves or intercalate themselves into the yolk sac, and that creates the opening, the neuroenteric canal. So at this stage in this illustration, the neuroenteric canal is not present. You would have to see the entire continuity from yolk sac cavity, if that's what I want to say first all the time, to the amniotic cavity. Thus, the oropharyngeal. Oh, question, yes.
Oh, I was just going to. Number 7 was body stock.
Yes. No. It's the allantois within the body stalk. Sorry. The arrow is actually ending at the extension of the endoderm lining the secondary yolk sac, and it is the allantois, A-L-L-A-N-T-O-I-S. The oropharyngeal membrane is another term. I use the term buccopharyngeal membrane, oropharyngeal membrane. The buccal cavity, of course, is the oral cavity. Same words for the same body cavity in the fully developed structure. That membrane will not actually form the mouth. It will close off the junction of the oral cavity or buccal cavity with the oral pharynx, the first part of the pharynx, just immediately caudal or dorsal, in the adult, to the oral cavity. We know that there's some pharynx that is superior to this. Right? What's the portion of the pharynx that is superior to the oral pharynx? Nasopharynx. Very good. So the nasopharynx is above this, and of course the nasopharynx opens directly into, other than the nasal cavity, directly into the oral pharynx, down into the laryngopharynx, and ultimately both into the trachea, through the larynx and the esophagus, the esophagopharyngeal junction. There is a cardiogenic area that we haven't pointed out, but I mentioned at one point that the migrating mesodermal cells will invade every location except prochordal plate and the future cloacal membrane between the the ectoderm and the endoderm, and those obviously doesn't get interposed between the epiblast and hypoblast cells of the prochordal plate and the future cloacal membrane. And that mesoderm that surrounds the prochordal plate, rostrally and laterally, it surrounds it actually as a halo, but the horseshoe-shaped or U-shaped collection just immediately around or at the perimeter of the prochordal plate will begin to differentiate into the primitive heart. So therefore it's called the cardiogenic area. So it is a U-shaped collection of mesodermal cells that are already somewhat specialized. They're a little farther along in their differentiation toward becoming what type of cell? The heart's going to pump because of what kind of cell that it has. Cardiomyocyte, or the cardiac muscle cell. Okay. Remember, mesoderm becomes all types of muscle. Skeletal, smooth, and cardiac. So those are the parenchymal cells of the heart, anyway. What will contribute to the formation of the stroma of the heart? Connective tissue structure. Mesodermal cells, right? And the vasculature of the heart? Mesodermal cells, okay. We're doing well. The cloacal membrane is basically same discussion as we had before, and we already pointed out the allantoi, or allantois, however you want to pronounce it. It is a diverticulum. We saw that extension, kind of like you'd push your finger from the inside of a balloon toward the outside, remove your finger, and that shape would remain. That's a diverticulum. And it is extremely important. I was mistaken in or I misstated that we don't understand the function of it in a species other than the avian. I was more thinking in reference to any type of urinary excretion function or a parallel of that. This will be a site of early blood cell development. And it will, in some fashion, in a sense more so a placeholder for the production of or formation of the urinary bladder, but it doesn't completely become incorporated into the urinary bladder. Well, that was lecture 7, sorry. I stated we were going to finish 6 and then go to 7, but it's finish 7 and then go to 8. Thus, we have the notochordal process that has sent a message to the overlying ectoderm, the immediately overlying ectoderm, and it will respond by beginning to differentiate into the neural ectoderm. Thus, we have a delineation of that ectoderm that doesn't go in that line of direction becomes the surface ectoderm. But we also have cells growing quite massively in plate, and when we get this plate-like flat displacement of cells in a radial fashion in this embryonic disc, then that purse-like constriction of the junction of the amnion and the yolk sac is going to force folding. It has to ripple. There's no place for it to go. It can't just become this gigantic flattened disc. So therefore, we're going to begin seeing some foldings and premature or primitive, let me say, body cavities that can be recognized. And we know what body cavities they will become. And following these body cavities will be one of the greatest challenges in trying to visualize the three-dimensional development of the embryo. You can think of a butterfly with its wings out, and if you pulled the butterfly wings forward, curling the tips so that they would meet ultimately, it obviously would have just one big cavity contained by the wings once they closed off all the way around. But it doesn't stay that simple. There's structures that grow into it and push to the side, make contact with the edge, so this space becomes three-dimensionally contorted and twisted, and so there's going to be canals, pleuroperitoneal canals, and all these other types of new spaces created surrounding the organ systems undergoing development, such as circulatory, respiratory, and digestive, until ultimately you end up with the, in some cases, somewhat simple, but in other cases, like the peritoneal cavity, complex, complete organism body cavities. The cardiovascular system obviously is essential. We've said that this structure is growing and no longer can rely upon simple diffusion for the source of nutrients and excretion, ultimately, of wastes. So the neural tube will begin to develop within the 3rd week, and obviously is going to continue throughout. So we have stated that the notochord is responsible for this. It will give rise to the central nervous system and the retina. It will continue to develop into a groove. So this is some of the first rippling. It's not just a rippling like if you were pushing some aluminum foil toward itself and it formed a nice uniform dip. There's also thickening of the cells on the margin of that. So you get a groove with folds that are accentuated by thickening or an increase in the height of these columnar cells. So they go from mid-range columnar height to tall columnar cells, accentuating the folds at the edge. And ultimately, there will be some cells that will migrate away from that junction of the the neural fold and the surface ectoderm, as we will see, to become the precursor of the peripheral nervous system. And when we proceed to that discussion of the formation of the central nervous system and the peripheral nervous system, we have to remind ourselves that if you were to take a peripheral nerve and dissect out a single axon, that could either be originating from a neuron cell body in a ganglion within the peripheral nervous system, or it could be the axon of a neuron within the central nervous system. So when we talk about the development of the peripheral nervous system, we're talking about the development of the sensory neurons that are located in the peripheral nervous system, and their dendrite, let's call it. We know it's more like an axon, but their sensory input process, called the peripheral process, is located within all the peripheral nerves, or all the peripheral nerves have those in them. There's the Schwann cells, there's connective tissue surroundings, and the vasculature. That really is the peripheral nervous system in the sense that it develops from these cells called the neural crest cells. But the axons of the neurons of the central nervous system from the neural fold cells and neural groove cells will extend their axon into that container produced by the structures we just described. The neural tube ultimately will be generated as we keep pushing our aluminum foil. Say it's between two tables, so our little depression can sink down in between the edges of the two tables, and eventually we can just push the two margins right together, and of course they're cells, so they will just form tight junctions and close it off into a tube, and then ultimately that's just going to separate from the rest of the aluminum foil that we had, and the aluminum foil fuses itself together, separates into a tube or cylindrical structure that we created, and then the surface is closed off. So we use the other analogy of the butterfly wings coming together and the edges of the butterfly wings all fusing together. So we will have a lot of this occurring with the margins of structures approaching each other, contacting each other, and then the cells just say, hey, let's form tight junctions and close this right off. So it's not really that magical of a thing, but it is really quite marvelous. By the end of the 4th week, so in other words, within a week and a half, we have the neural tube, and the neural crest cells, as we'll see, will be there, and the framework for the central and the peripheral nervous system is there. So why do we have to have the nervous system developing so early? We understand the circulatory system, but why do we need the nervous system developing so early? What if the upper limb developed first? How would the developing nervous system, central and peripheral fibers that have to get out to all their destinations get there? Right? So they're going to develop, send out their processes, find their general location, a lot of chemical communication, and then as the cells, their target cells, get displaced or migrate, whatever the process, to their final destination, the axon or the dendrite of the sensory fiber just continues to extend and lengthen to reach that final destination site too. So we've got to get the nervous system there first. It's not really going to be functional and coordinate things for quite a while, but we've got to have it in place so that it establishes its friendship with its target cells and sustains that. So this is the same view that we looked at before as the neuroenteric canal is being produced, where the notochordal process opens up with the yolk sac that would, of course, extend back. That section is, transverse section is at this level. If we went farther back, we could follow this right up through the neuroenteric canal. Well, we saw this before. The overlying cells are already starting to thicken, more so increasing in their height, and we already see a groove being established by the accentuation of the margins, the tall columnar cells. So these are the neural folds, and that's the neural groove in between the right and left neural folds. And this becomes more and more well established over time. Thus, we can go again to the Gray's anatomy and look at this really quite marvelous illustration of the human embryo late in the 3rd week, where we recognize the primitive pit, as we were referring to it in the other illustration, the primitive streak. The future cloacal membrane would be way back here. So obviously we have had quite a significant growth in the more rostral portion of the embryo. And this was true. The rostral end or cephalad end of the embryo and fetus will develop more rapidly than the caudal end. Therefore, hip dysplasias or incompletely developed lower body structures are more common than rostrally. That's an unsafe generalization, but it still is just the same. So we orient to the cut edge of the amnion, and we therefore know we're looking at a dorsal view of this. We identified the primitive pit. Now would be open into the neuroturic canal, so we know the notochordal process extended rostrally to the prochordal plate. Therefore, the overlying cells reacted, a heightened or an increased height or tallness of the margin cells on each side, generating this groove in between that is an accentuated surface feature right now, but ultimately becomes a well-formed fold on each side and groove down the middle, and ultimately these edges will approximate each other and close off. So this is the neural groove. We see the body stalk connected to the chorion, very well established here in this view. And this is the illustration from our textbook. Basically the same thing, identifying similar structures, but it now is pointing out the cardiogenic area way forward of the prochordal plate. We know that the neural folds and the neural groove cannot extend rostral beyond the prochordal plate. Why? Why is it that the neural folds and neural groove cannot develop at or rostral to the prochordal plate? The notochord could not get into the prochordal plate nor pass in front of it. So the prochordal plate would be right at the tip. The mesoderm that is forming the U-shaped structure around it is going to be that cardiogenic area. So the neural plate is another term for these thickened cells before the groove becomes accentuated with the establishment of the neural fold. There are some spaces actually beginning to develop in the extra, would be in the extra embryonic mesoderm, and these are becoming some of the coelomic spaces. So we'll follow that terminology. There'll be intraembryonic and extraembryonic, and it gets a bit confusing at times with the inside the embryo and outside the embryo. Again, the cut edge of the amnion telling us it's a dorsal view, and the body stalk shown here. Thus, we can take a closer look at the embryonic disc in this transverse section. We see the notochord now is cylindrical in shape, but is not a solid core. The mesoderm that extends to the right and the left, let's say, laterally from that midline structure, are divided into three portions: the paraxial mesoderm, intermediate mesoderm, and the lateral mesoderm. And those coelomic spaces we saw from the dorsal view are forming within the lateral mesoderm intraembryonic. So are these empty spaces? What would we find in these spaces? Extracellular fluid, right? So there's never just an air pocket that's going to be developed. There will be, just like the formation of the antrum in the developing follicle, there would be the extracellular fluid accumulation separating the cells, same as the formation of the blastocyst, extracellular fluid accumulating, separating the cells. Here, the same thing, extracellular fluid accumulating, separating the mesodermal cells, forming these coelomic spaces. And obviously, the ectoderm dorsal to the notochord on either side of it, running parallel to it, has become much taller, accentuating that area, but also putting a little pressure on it, so a little indentation, forming the neural groove with the neural folds on each side. Just before this occurs, where you get the accentuation of the margins and the beginning of the groove, you get just a generalized increase in height of all of these cells, and that's that neural plate that we saw labeled in a previous one. So the neural plate ultimately will have these margins on the side. The cells get much taller, forming the folds. It'll put a little pinch on the inside, the midline structure, and not only displace it ventrally, but also just be taller cells, so we get the groove and the folds on each side. At the edge of the fold, I would actually have, well, they're just labeling all of this as the embryonic ectoderm. But now that we have the neural fold, there is a margin somewhere here where this is surface ectoderm and this is neural ectoderm. So the blue, red, and yellow obviously are the standard colors within our textbook and in many cases for ectoderm, mesoderm, and endoderm. Well, already we're beginning to see that the terminology is layered. We are introducing a variety of terms, paraxial, intermediate, and lateral mesoderm. Ultimately, we're going to follow those through the entire lineage of production of different cell types, organs, and organ systems and tissues. So it pays to lock these terms into our memory as best as we can now, because they'll come back to visit us repeatedly through the remainder of the discussions. As we proceed into the fourth week, these coelomic spaces are becoming larger. There's more extracellular fluid collecting, beginning to expand in all directions, and we can predict what's going to happen. They're all going to fuse together ultimately and form some type of a ring-shaped structure, another U-shaped structure, horseshoe-shaped, extending from the lateral caudal edge rostrally to the midline and then mirrored on the opposite or contralateral side. We see that there are some other structures beginning to develop at this point where the neural fold and neural groove become quite accentuated, especially rostrally. And this structure is called a somite. So the underlying mesoderm is undergoing some changes. We recognize the SOM as being from the suffix soma. So these are beginning to lay down some of the body structures. And just, this is global body in general. So these are going to show up. Obviously, if this is the first one, there's going to be multiple ones. So we are going to lay down first the segmental orientation from head to toe, if you will. And we know that we are not segmented organisms, although some organisms do persist as segmental. So somehow these are going to have to interact and fuse to form one continuous organism that can't be pulled apart and have unique or complete, basically, sets of organ systems within them, such as the earthworm. So this first somite becomes a point of orientation. So we'll begin, and they're not even labeling it anymore, we'll begin kind of ignoring the Narantura Canal or the primitive pit and the primitive streak. As soon as we have a somite, that gives us, the first somite, that gives us rostral events and caudal events from that point of orientation. And, the continuation through the third and into the fourth week is really just a more complete establishment of all of these structures. The somite is generated by a tremendous change, obviously an increase in the number of mesodermal cells within the paraxial mesoderm, and that creates an elevation in the overlying ectoderm that is visualized and called the somite. So the intraembryonic coelom has developed as we had described. Remember, we're going to be using these terms intraembryonic and extraembryonic quite a bit now. As soon as that coelom is quite well established, we have a fluid lake, if you will, a fluid-filled potential anatomical space separating the mesoderm, the lateral mesoderm in those areas. So we need new names. So we have the intraembryonic somatic mesoderm adjacent or subjacent to the surface ectoderm, and then the intraembryonic splanchnic mesoderm superjacent, I guess we would say, superjacent to the endoderm. And now we see this continuous U-shaped intraembryonic coelom taking on new terminology. That space near the cardiogenic area, the pericardial coelom. So remember, we said the neural plate and groove cannot extend past rostral, past the prochordal plate. The mesoderm in that area is the cardiogenic area, going to become the cardiac myocytes and connective tissue and stuff. So the space that the intraembryonic coelomic space that surrounds that area is pericardial. Then we know that the abdominal cavity way down back here ultimately is going to be an abdomen, right? Is referred to as the peritoneal cavity in the adult. So there is a continuation from the pericardial to the peritoneal. We know lungs are going to come up in here someplace in between, but for now it's just a continuous fluid-filled cavity from the pericardial all the way to the future peritoneal, if we will allow ourselves to say future. And the continuity between those is the pericardio-peritoneal canal. This is the space, the pericardio-peritoneal canal, that's going to become very distorted and pinched off and separated into a bunch of different other spaces as the heart develops and we see all of the folding. So I'll stop with this slide as you're picking up. We're seeing the fusion of the neural folds, or the proximation of the neural folds, and their free margins will ultimately fuse together, and we can just picture these cells taking on connection with each other, losing connection with the remaining ectodermal cells, and then just mesoderm interposing itself between the surface ectoderm and the neural tube, and we have that complete neural tube. Appreciate your time and attention. I'll see you on Monday. I'll just make the quick statement now, but I'll reinforce it on Monday. We will not show up in class on Wednesday. Why? Because you're all going to go to the Health Professions Fair from 10 to 2 in the UC Center, right? And then 9 to 10, you're free to come over and help out, just in case we don't have enough volunteers.
Monday we take the test, right? Are we still going to do that?
What's that?
This Monday.
Well, the test is online.
Right, so
We're
we're still
talking
going
class
to.
on Monday, right?
Yeah, there is lecture on Monday.
You
Yeah.
said Wednesday we don't have to come to class.
Wednesday is where I won't even come here because I'll be over there.
You're going to be at the health fair, right? During this class period?
Yeah, from about 6:30 a.m. until about 3:30 in the afternoon.
And then on the test, you say we have 2 chances to take it?
Yeah.
And after the first time that we take it, do you tell us the ones that we got wrong?
You get to view that.
And it tells you which numbers are wrong?
Yeah, you don't get to see the questions.
Right.
But you have permission to have a pencil and paper while taking the exam online and just kind of making notes. Question one. There obviously isn't time to write down the whole question, but you can write little
let you know
hints so you can go and study.
So it'll say like
Clarify
number
it, come
13
back.
is wrong, and
Right.
if I have a little note, I can keep it in my mind. So got it.
Keeping it in our mind is the important thing. That's
Right.
learning, right?